sell education and information. It's definitely not as sexy as selling like the natural whatever.
How are people not more afraid of being stupid? Because that's what they should be afraid of.
Can we meme this quote, Dan?
I mean, we are recording, and I just hit live on YouTube.
That is my favorite, one of my favorite things anyone's ever said on this show, because that is perfect.
Yes.
That's so good.
Oh, so I think we just jumped right into it, right? I don't know if we wanted to.
Yeah,
we do a little bonus content here and there, because as we were talking about monetization, we have a Patreon that we like to have some bonus stuff for. So I'm gonna put on Facebook later today. All right, so I'm gonna do the intro, and then we will actually start what will be the content of the podcast. All right. As soon as I hit live on Facebook, which I did, but there's like a 5 to 10 second delay.
Okay.
So, I mean, it all depends.
All right. Well, I'll just get started. Tonight, we are thrilled to be joined by Dr. Lynn Curchoe. Carol is an American reproductive biologist specializing in molecular biology, cell biology, and biotechnology. Her key contributions to those fields include advances in stem cell culture, epigenetics, and reprogramming. She is the former Utah State Science Advisor, Domestic Outreach Director of We Love GMOs and Vaccines, and is an author of personal essays and fiction. So welcome, Carol, to our show.
Thank you. Glad to be here.
So before we get into this episode, which we may or may not be calling something Dan came up with a couple months ago.
What the fuck is CRISPR?
Yeah, because this is our question that we really have for you, is what the fuck is CRISPR? We want to first get your sort of scientist origin story. What brought you to science as a career?
Yeah, so I always loved science growing up. I always knew I was going to be a scientist, but I actually did come to it through a non-traditional route. I was a high school dropout. So I went to community college. I have a GED. I transferred to the University of Connecticut. And all the while I had fantastic mentors. I think, eventually I thought I'm just going to be a technician. I'm going to get my degree in biology, biotechnology. I'm going to get out of school. I'm going to get a job. That was my main priority in life. But I had been working in a lab to get some of my undergraduate credits. And my advisor said to me, what are you going to do with the rest of your life? And I said, I'm going to get a job. And she said, you don't know this now, but you need to get a PhD. And I didn't even know what grad school was at the time. All I knew was that grad students didn't tend to like the undergrads in the lab. They were these mysterious, shadowy figures that were supposed to be mentoring us, but didn't. And so then my teacher helped me to get into grad school in her program. I was very lucky to have a full funded scholarship, probably on your taxpayers' dollars. So I hope that everybody's happy with the contributions I've made to humanity and what your taxpayer dollars were used for. So I worked in a lab, the highest biotech lab on campus. We actually cloned cattle. So that launched my career directly into the stratosphere of controversial biotechnologies. The very first study I was involved with was the study that basically proved that meat and milk from cloned animals were not any different than meat and milk from traditionally bred animals. So I've been pretty much in the public eye ever since on various things. So cloning, human embryonic stem cells, and biotech of all sorts, using the system, using components from cells, which was a little controversial as well. And now I just do private embryology.
Wow, so you really were just right in the midst of things that probably a lot of people do consider controversial.
Yes.
Right from the beginning.
Absolutely.
Did you ever have any hesitation with any of the topics that you were delving into or working on?
That's a good question. So, of course, when I first started grad school, I was very hesitant to we work on animal models in the lab. So there is a certain number of sacrifices that need to be made for every project. But luckily, I was with a very lab ethically. University of Connecticut is very highly regarded with their bioethics, and every study I was ever involved in, we ended up running through the proper IRBs and the proper channels. But it was hard at first because I did have to learn how to humanely sacrifice animals for research studies. That was probably the toughest part. I had taken a number of philosophy classes as an undergrad, and so I was pretty well prepared for the arguments for and against technology. And since I really strongly believe that everything I've ever done has been toward some greater end for the I think I've always felt, okay in what I was doing.
Yeah, that's cool. Yeah. So, I mean, because you're obviously working in a field where there's so much that's been done, but so much on the horizon, hopefully, in the world of biotech and all of that. And the reason we wanted to have you on was to talk about one of the, I guess, big things happening, which is CRISPR, which I think when there were a lot of articles going around about allergy-free peanuts and that kind of thing was my first, I guess, introduction to seeing CRISPR in all caps kind of put in articles and stuff.
Sure. Yeah. So the allergy-free peanut is an intriguing use of genetic engineering. I think that since it's a gene trait and it's an interaction between organisms, ourselves, and other organisms, plants, it's probably going to be pretty difficult to actually pull off in practice. I think there's probably something environmentally that we need to get under control first, as far as where these allergies are coming from. What are we doing? I know my pediatrician has me exposing my infant daughter to very small amounts of peanut butter to kind of inoculate her against those allergies since the time she was 4 months old. So, but yeah, CRISPR. So CRISPR is a fascinating new tool in the genetic engineering toolbox. It's very easy to use. It's very inexpensive. And we're teaching biotech to 7th graders now. So kids that are very young are using CRISPR in their science fair projects. Wow. It has become incredibly easy to genetically modify organisms. CRISPR has the potential to turn any organism into a model organism. Bacteria to yeast, which we already had, all the way through mice, all the way through rats, which we already had, but to some of your more advanced organisms, it's been very hard to genetically engineer monkeys and primates. It's been very difficult to genetically engineer other animals for agricultural uses. So now, for example, they're making a cow breed variety that doesn't have horns. So that will be really great for cattle. It'll be really great for the farmers that have to do the dehorning of the cattle. It'll be really great to reduce animal suffering in and of itself.
Can they edit it so it makes chocolate milk instead of regular milk?
What about strawberry? What about strawberry milk?
Yeah, Dan. There's so many other
all
flavors.
For it.
So many other flavors out there, not just chocolate. But yes.
What about strawberry chocolate?
Oh, that's it.
I actually think they could do strawberry. Because
she's like, there's something here. There's something here.
Right.
There's
you could have it like a strawberry. Yeah, we could just have strawberries growing on the backs of cows, and then.
No, the flavor molecule for strawberry has been pretty well worked out in the lab, and in the food labs in New Jersey. I think they know the sequence for that. I think chocolate is actually a pretty complex flavor.
So start small. Start with strawberry.
Yeah, exactly.
These are the things that can change the world. This is how science will change the world. So yeah.
Yeah.
I mean, that's how to get people, or at least one way.
Yeah, absolutely.
And get a lot of bad science articles written in the meantime.
Yeah. And I think one of the really exciting applications of CRISPR is making these consumer products that people kind of don't know about yet. So there's been, for example, a really famous, this wasn't done with CRISPR, it was done with other genetic engineering techniques, but a very famous researcher out of Utah State University who is producing spider silk and goat milk. So spider silk proteins in goat milk. And that's, it's, you know, spider silk, it can't be made synthetically. So being able to biofarm that is huge. It has incredible tensile strength. And when you start to move something like that from these small organisms like spiders that you can't really farm and that you might be destroying their habitat to an animal that we use already for agricultural purposes like a goat, you are really opening up the doors to preserving habitat, better products, and, you know, just in general, you're sort of lessening the footprint of humanity. Want to exploit those natural resources. And really in some cases, it might be better to genetically engineer them. Yeah. So, but I think we kind of jumped over the what the fuck is CRISPR?
Yeah. I figured, like, let's just explain it to us, like like people who don't know.
I heard strawberry cows, and
he went with it. He went,
That was just.
All viruses like to inject their DNA into living cells. Viruses are basically a paragon of Darwinian evolution. They've jettisoned every single unnecessary aspect for life, and they're basically just DNA syringes or RNA syringes. When they inject their DNA or RNA into your cells, they don't actually have any replication machinery of their own. So their DNA integrates into your genome, and then your cells start pumping out new flu particles. That's how viruses work, right? Mm-hmm. So a very rudimentary organism is called a phage, and a phage is a virus for bacteria. So single cell organisms like bacteria get viral infections as well. And bacteria cells have a very rudimentary form of an immune system. So the researchers that discovered CRISPR were actually working on bacterial immunity to viruses and what that looked like. What does the bacterial immune system look like? And they discovered these repeating sections in the genome of the bacteria. In between those sections of repeated DNA, there were small segments of viral DNA up in the bacterial genome. And so the researchers basically discovered that this was the way the bacteria had been infected with a virus. And then it could mobilize its resources to fight off the virus quicker. It's exactly like our immune system. It's called the adaptive immune system. It means you're exposed to something, your body remembers it, you fight it off. So some very clever researchers decided to exploit the natural machinery of the bacteria to insert DNA into an organism. And so what they use is an enzyme called Cas9. So the enzyme is the actual molecular scissors that cleaves the DNA. Then they have other sorts of clever techniques for either making the cleaved DNA simply join back together with itself, which is called non-homologous end joining for any really nerdy types out there, or inserting a brand new piece of DNA with a corrected sequence. For example, if you're looking at a single gene disorder that has a single nucleotide base that needs to be repaired, or an entirely new gene itself. And then once that's in the DNA of the organism, the cells are going to replicate, it's going to make that protein, and it's going to have that trait. Does that make sense?
Maybe.
Yes. Maybe.
Yeah. But well, I think that what's amazing to me is, as you're explaining, you also said before that being used and taught seventh graders.
Mm-hmm.
And that's kind of amazing to me because I wasn't doing that in seventh grade. No,
yeah,
not at all.
This is actually kind of interesting because it's intertwined with what we call the next generation science standards in this country. What are we teaching seventh graders and eighth graders? And genetic engineering and biotechnology and working with DNA really has, in some aspects, moved down to the level of the technician. So when I was in my graduate school program, sequencing a new gene, you could write a PhD thesis on. But now, really, it's become automated by the technologies we have today. So these kids, you know, can make some primer sequences and, you know, all the tools for these things are online and they really can understand how to do this. So you can buy a PCR machine on eBay. You could do this in your garage if you wanted to.
Is that sort of like the biohacking type of thing?
Yes.
That's a term that I've heard come up. So does this fit in with that idea of biohacking, citizen science, and just people using it, trying to work with the technology?
Yeah, it sure does. There's a humongous community of what is called DIY biologists who are doing biology in incubator spaces. They're doing it in their garages. They're doing it in communities online. And they're getting results in some cases where the establishment has moved very slowly. So the advent of crowdsourcing information and crowdsourcing data and being able to connect with hundreds of thousands of people all over the world has actually led to some advances in rare disease research where people have been able to pool their resources, do genetic sequencing, and find the mutation responsible for some rare diseases. I mean, it's amazing, really.
What's an example of a disease like that there's been research done that, or I guess garage research done that's contributed like that?
Yeah, so I don't remember what the actual disease was, but I think it was something very rare, and it was a father whose daughter had it. He had no background whatsoever in biology, and basically assembled a community of people where everybody had these similar symptoms. So they put sources together and sequenced everybody's whole genomes, compared them together, and found the mutation. I can't remember what the disease is now. Sorry, Dan.
It's okay. We'll forgive you.
So, kind of along the same lines, something that our friend Jason brought up when he and I were kind of chatting a little bit today about us interviewing you, because this technology is perhaps cheaper, easier to use in some ways than other technology. He kind of was asking, how does this impact scientists or citizen scientists in the developing world who have less access to, big lab resources? Is it possible to make more advances because of this?
Yeah, I think we're going to see an explosion in things that are coming out of the developed world. And I think that it's going to upend the, silos and the ivory towers that we have. I mean, we've already seen new crops being made in third world countries, and the first genetically modified embryos came out of China. Recently, the US just published a research article with human embryos as well. But I think you're definitely going to see that this is the great biology.
So there's exciting stuff on the horizon, hopefully.
Yes, very exciting stuff. And I think the last year, the government agency that regulates crops examined the case of this non-browning mushroom that was made. And since the CRISPR technique is just delivering an enzyme, and in the case of the non-browning mushroom, it didn't add a gene, but it just disrupted a gene. So essentially it turned a gene off. It wasn't adding anything. And since it's not using a virus, like the traditional methods of crop modification, the regulatory agency basically said they're not going to regulate it. So GMOs in our country are regulated by the method that's used to produce them. And since viruses have this neat trick of inserting their DNA into plants as well as animal cells, the traditional methods of genetic modification have relied on viruses. And, viruses can be considered pests. They certainly have developed their own evolutionary mechanisms to annoy us in a lot of ways. So no matter what the scientists really did who used those old techniques, they did all kinds of things where they cut out every sign of the viral footprint out of the DNA, and they did all sorts of tricks. The regulatory agencies never liked these genetic engineering methods that use the plant pathogen. So they do like CRISPR because, this enzyme is actually present in any bacteria. It's present in all the bacteria in your body. It's present in every bacteria that you come across on any surface in your house. So there's plenty of Cas9 to go around, I guess is the. And they particularly like it when you're just shutting off a gene and you're not adding a gene from another species. So they liked the case of the CRISPR mushrooms, and I think those will be available on the market within like a year or so. But now that kind of brings us to, well, the first paper was just published in Nature of U.S. scientists genetically modifying human embryos, a single gene disorder, and it was a cardiovascular, a cardiomyopathy. To correct the defect in the DNA of the embryos when they were just a single cell. So then as the embryos grew and divided, the change propagated to every cell in the embryo, which has traditionally been the problem with genetic engineering. You can't deliver it to every cell at once. You need to sort of target embryos when they're a single cell. And there's only one genome you need to modify.
And wasn't that something where they had 9 single cell embryos and it worked in like 6 of them?
Yep, that's the one I'm talking about.
And then of course they none of those they destroyed all the embryos after that, so there's not going to be CRISPR people
right. Yeah, it's really interesting because so now I work in an IVF clinic making babies for people, which is awesome. It's very fulfilling. I love doing it. That's one of those memes, like, explain your job poorly. I make babies for people. Yeah, I make babies for people. So which I love being able to apply my science to, a very direct way where I'm helping people every single day. But it's pretty highly unregulated.
Right.
I mean, we can take techniques directly from the research lab and apply them to the production of human embryos for people. And there's really not a lot of regulation. So one of the things that I do on a regular basis is biopsy embryos and send them for genetic testing. As a woman gets older, her eggs have been sitting in her ovaries her whole entire life. A lot of people don't realize this, but meiosis, or cell division in the egg, finish happening until the sperm has penetrated the egg. So in sperm, all the meiosis has finished happening before the sperm is ejaculated. For the egg, the last meiotic division is not going to happen until after the sperm penetrates the egg. And so if people remember their mitosis from 8th grade biology, the chromosomes line up in the egg, the spindle fibers come in, they grab the chromosome by the middle, and they drag them apart into the cell, the mother cell and the daughter cell. So as a woman gets older, it's really difficult for those spindle fibers. They get crunchy, they get old. They're 40 years old. They've been sitting in your ovaries, and now you're waking them up and expecting them to work right. And often they break and leave a chromosome behind, which is why you get trisomies, Down syndrome, trisomy. So we like to test we like to use genetic sequencing to test a few cells out of each embryo to make sure that they have a normal complement of chromosomes. And that also typically tells us whether there's an X or a Y chromosome as well. So we do a lot of gender selection, which we affectionately call gender balancing. Okay.
That's good.
And yeah. And you know, I think this brings us back to Jason's question of the ethics. Is what we're doing ethical?
Yeah.
You know, can a lot of people access these technologies? What happens if a lot of people do access these technologies? You know, we do thousands of IVF cycles a year.
Yeah.
I mean, we're making babies for people who otherwise would not be able to have them. So that's another interesting thing as well. Are we allowing genomes to propagate? You know, the question of, are we allowing these genomes to propagate that normally would be kind of weeded out because they wouldn't be able to reproduce naturally?
That's an interesting
so
question.
All of those,
yeah,
I know, right?
Yeah.
So all of these questions, as well as, you know, there is a bit higher of an instance of embryos that have been cultured of, you know, diseases, being overweight, certain genetic abnormalities, simply from being in culture inside a lab before getting put into a person. Because you have to think, like, embryos don't have any mechanisms that a human body has. Like, they don't have any pigment, they're not protected from UV, they don't have any lungs, they can't regulate their own pH, they don't have any, you know, they can't regulate their blood chemistry, as it were. They don't have any blood, you know, capillaries or blood cells of their own yet. So we have to do things to really protect those embryos before we put them into people. But sorry, I don't know where this is going. What are we talking about here?
Well, but it shows how complicated it is, though, too. Like when you take it, or I guess how big the discussion can get, in a sense, when you take it to talking about humans, because you're not just talking about food, and you're not. Yeah, it's people. I mean,
in other words, we're not doing that level of things in my garage, at least.
Right. And so I think it's interesting because I did a sabbatical in Boston. Okay. And what I discovered while I was there is that in the state of Massachusetts, the insurers are required to cover infertility treatment. So if you are infertile and you've been diagnosed with that, you can go to the infertility clinic and get treated as part of basic human rights to healthcare in the state of Massachusetts. So that's really interesting because the state has required insurers to say, you know, infertility is a painful enough disease that we expect you to cover this, or you're not going to offer insurance in our state. You have to cover this. So it is kind of interesting because at least in Massachusetts, as one of the states, it is very democratic whether you are poor or whether you are rich. You can access infertility treatment.
Right, because in other places, then it would be just if you had the means to do it, right?
Yeah,
because
exactly.
Because I guess I kind of forgot about that part of it too, is that it is expensive, right?
Yes, it's very
go
expensive
through this process if you are not sort of guaranteed that, like you said, in Massachusetts, which I didn't know before. So
yeah, and I think, you know, as we as a country examine our attitudes toward people's access to basic healthcare and what should be included and what shouldn't be included, you know, I think it's really easy for the president to send out a tweet about transgender in the military because of the cost of insurance and get everybody fighting with each other about what is and isn't covered, when the real problem is none of us have access to great insurance.
Oh, yeah.
So then we start fighting, in group fighting. Oh, transgender surgery shouldn't be covered because I couldn't get my LASIK eye surgery. Right? Which is an actual argument that I heard.
Really?
So, that's a real
ignoring the fact that it's already not.
Yeah.
It's an elective, or considered an elective, I should say, and so it's already not something that they have to pay for in the first place.
Well, you know, it's just I mean, let's just put it this way. People want to argue morally what should and shouldn't be covered by insurance, but that's not actually the right argument to be having. The right argument to be having is, does a doctor say that you need this, and does it improve your quality of life? Because our founding documents saying life, liberty, and the pursuit of happiness, we are all entitled to those as human rights.
But somewhere that got lost by our
government. Because
they were drunk
human.
when they wrote that. So
our
they
government does not
really
give us those rights. Those rights are ours because we're human. They cannot be given to us by politicians.
And I may be wrong in this.
But
I think we're lagging a little bit. I may be wrong in this, but I believe this happened with what was that? The 11-month-old that had the what in Great Britain, what they consider to be
Charlie Gard.
Charlie Gard, yeah, with that child.
Yeah.
And it's my understanding, at least in Great Britain, that the doctors always have the final, or there's some sort of like override over what the patient wants. Have you heard that? Am I recalling that correctly, or am I just completely making things up right now?
So, I mean, we have that here too. Doctors can refuse to do unnecessary and potentially damaging treatments, and doctors,
right. The way it was presented to me,
routinely.
The way it was presented to me was a we don't want to do this. We think this is wrong, but physicians say, well, this is what the research tells us. This is what we need to do. We have every indicator that we need this treatment or we don't need that treatment, and then that's it. There's no I'm gonna doctor shop and find something else. That's just the way it is. Is the actual medical professional has the final say. Is that because I'm rather ignorant of a lot of things, including this.
Yeah, I mean, I'm not an expert on that case by any means at all, but the way I understood the case is that it's not that the doctors were refusing to do it, but it's, you can't force doctors to do something from the research literature that's not established medical practice, right? Like, say you were just in this country, like say you, Dan, you have cancer, and you read somewhere or in a research study that you wanted a whopping dose of whatever vitamin administered to you, through an IV
B12.
With your turmeric,
not B12, whichever one is cyanide.
Yeah, cyanide. Yep. If every doctor that you approach in this country says, I cannot do an experiment on a human. I mean, it's just not ethical, right? So the way I understood that case is that the parents thought that there was something more the doctor should be doing, and the doctor said, we can't do that.
Gotcha.
Now, speaking... Yeah, we learned something. So, speaking to, again, to ethics and gene editing and all this kind of stuff, I was reading today an article in the New York Times about how people, just the idea of designer babies, and people have this fear that, with the possibility of gene editing, people who can afford to do so will be able to essentially,
create a master race.
Yeah.
Create the little masterpiece children. So can you kind of speak to, I guess, the fear associated with that some people have?
So in order to change a trait in a population, you would have to have a tremendous selective pressure that changes every organism essentially at once in a population. So if you're just looking at genetics from the foundational study, the basic study of genetics, and under normal circumstances, without genetic engineering, you wanted to change a gene everywhere in the population, it would be almost impossible to do without a really severe bottleneck, basically restricting the whole population down to a few founders that had that trait. So actually, this is what you see in the negative way in some populations. So I'm French Canadian by descent. My family was. But in French Canada, we have a very severe effect called founder effect. There were only about 80 sires that came over from France to populate the new French colony. And they were instructed to have very large families, which, I'm one of them. My grandmother is one of 15, and each one of them had at least 5. And my mom I'm one of 5 kids,
Yeah.
But it's a very small geographic area. There was a lot of inbreeding. There's a lot of inbred genetic diseases because of this bottleneck. So we have a lot of Tay-Sachs. So do Ashkenazi Jews. We have a lot of cystic fibrosis. And we have some really weird things that come up, like there's a syndrome where the muscle in the eyelid fails as women get older in French Canada. They need to get a sling inserted into their eyelid to pick it back up again because they literally can't, open their eyes wide anymore, right? So it's really bizarre, but it's like a connective tissue disorder of the eyelid. And it's like, where do you see that except in an inbred population, right? So
what I'm hearing you say is Canada is a bunch of inbreds. Is that?
Yes. No, French
That's going to be the one thing people take away from this conversation.
Yeah, no. And you also see that in sort of other populations that have had a forced bottleneck for whatever reason. So the Ashkenazi Jews, like I mentioned, or there's, lots of other nationalities that for either reasons of geography or political segregation or whatever it was, they have had a severe genetic bottleneck. But, so basically to that point, as a master race to happen, well, human traits are multigenic and they're expressed in an environment. So not only would you have to control multiple traits, but then the environment that those master race people are raised in apply the same pressure to every organism in the environment. And that's just not possible. So if it were possible to do this, I think you would see that the Germans would have succeeded in having only very tall Germans. But height, as we know, is a multigenic trait. And so even if you have 2 very tall people breed together, you have regression to the mean, which is a genetic term, of their offspring. So offspring tend to go, even of 2 very tall individuals, will tend to go back toward the mean. So even if you could change it in 1 generation, you wouldn't be able to keep that change going in these traits that are multigenic, and they reach their potential in an environment. Yeah. So you can't really control the environment of an organism throughout its life.
So really, I mean, as
we don't want to,
like any of these things, it's probably like when people think about, and hopefully this changes more over time, when people think about gene editing or biotechnology or just the idea of genetic engineering, there's fears that just go along with, people saying playing God or that kind of thing, right?
All the way back to Greek mythology, where you see a fear of technology. So, there's the myth of Icarus fashioning wings out of wax and flying too close to the sun and the wings melt, and bad things happen to people when they try to use technology. Right?
Oh, that's fantastic. Like, I didn't really think of that story in that way, but right. It's true. It's true. He was doing the thing that was just pushing all of the limits, and when you fly too close to the sun, you're done. You can't do it.
Yeah, exactly.
Because it does get hotter the higher you go in the atmosphere too.
In the human psyche.
Like, don't push it too far.
Yeah. And I mean, personally, Jurassic Park is my favorite movie of all time. Because it has all the themes.
We could have a throwdown with get Trevor Valley back on here.
It has man's, hubris and man versus nature and nature versus nature, and it has everything in Jurassic. It's like the best. It's all the things.
Incorrect depictions of dinosaurs.
whatever. We're talking about some, but yes, that aspect of it, the man-made and then nature, and then it all fights back.
I learned something at the Children's Museum yesterday with my kids. They had a paleontologist there who said the velociraptors in Jurassic Park were actually closer to the size of a swan, which again, equally terrifying if you've ever encountered a swan or goose.
Swans actually can be terrifying.
Yeah, they're a little hard.
It was more their size, not instead of actually people size. So I learned something at something designed for 8-year-olds.
You're learning stuff all over the place, but but basically people are scared of sometimes advancing a little too far because it's advancing into the unknown. Yeah.
And that was something I wanted, whenever we were talking a little bit earlier, I wanted to ask about is you had mentioned that it's less regulated, or not less regulated, but with the mushroom, because you're turning off a gene, you're not adding anything to it. It's not going to be regulated. Like, just, I would like to know, as somebody who's actually in the field, like, what are your, like, how do you feel about that not being something that's not regulated, and is there a potential for that to be and I understand we're not adding anything, and that we're using a virus as the mechanism to deliver the change for, but is there a potential that we can turn off something or stop an expression of a gene that ends up having some sort of adverse effect?
So I love this question, Dan, because Americans love to be exceptional. We hate to look at what the rest of the world is doing and follow suit. We have to try to do our own thing. It's such a trait in America, but all we have to do is look north toward our neighbors again, Canada.
Well, they're a bunch of inbreds. We already talked about this.
The world. And so, in other countries, they regulate the end product that develops it. So I think what you're going to see here is we like to have knee-jerk reactions. We like to have lobbying, so we're definitely going to see that right now it feels a little underregulated. Change, the pendulum will swing too far in the opposite direction. It'll be overregulated and it'll be stifling. Then they'll back down on it. And in the end, we shouldn't be arguing about any of these production methods. We should just be regulating the end product. Is it safe? What did you do? What gene did you modify? What was the reason? Where are your safety documents and all of the testing for any product, regardless of how it was made? But I think, the interesting thing is about the current administration is they can be anti-science, but they're also anti-regulation. So we're already seeing the effects of that, because, the GMO label that was going to have to be on everything with the previous administration, that will not be enforced, I don't think, going forward. So all of the lobbying, of course, it was the GMO producing states that helped to get our current administration elected. So I don't think you're going to see that that's an enforceable item on the agenda of the current administration.
Current administration. That's a whole. That's like 500 other podcasts, because my goodness.
Use the word administration loosely.
Right,
I don't much administrative programs
I don't think the administration that wants to get rid of the EPA and the Department of Education. I don't think we have to. You're right. Don't have to. I had not thought of that.
Dismantle the FDA.
So now, all right, dismantle the FDA. So now we have a good thing.
Well, here we come back to the issue of how do you ensure that biotechnologies don't cause harm, and that companies don't take, on the mantle of biotech and start deploying things into the environment that will cause, some kind of cascading effect. And how do we regulate technologies in this country? And people really love to point to the few cases where something was supposed to be regulated, but somehow it got through, or you didn't see the effect until it was deployed in the population at large. You know, for example, there's the thalidomide. I love to talk about thalidomide, which is actually an example of it didn't happen in the USA because the FDA was smart enough to prevent it from coming here. All of those thalidomide babies were born in Europe. And, you know, but how did that get through? How is it that a racemic mixture of molecules could have an unintended effect, but the purified form, where it was only one form of the molecule, had no side effects at all, right? So it's, again, it just points to the need for regulation and having these frameworks in place that follow the scientific method. You know, it has to be evidence-based. We have to collect evidence that supports or denies the hypothesis, right? So that's, you know, why science education is so important to me, because I'm really hoping to, you know, make it so that, you know, the people that are going through school now and are exposed to these next generation science standards, and you know, the kids that we're educating, they understand that the scientific method is really our best way of knowing what is true in the world.
Well, I guess that kind of can lead into like a question I wanted to ask you from a more personal standpoint, because you, like you said before, you have adorable from all of her like, Facebook and Instagram pictures. She is so adorable. You have a fantastic little daughter. And I guess and this I posted on Facebook earlier today. I told my 4-year-old son, interviewing a scientist named Carol tonight, and he's like, "Is she a science mom?" And I said yes. And
so
you know,
cute.
and so yeah, kids are adorable sometimes when they're not, you know, having tantrums about snacks and stuff like that. But I wanted to ask you, as a parent doing the work that you do, you know, and keeping in mind that the work that all of us do now is, you know, setting the stage for future generations of people. Like, how does your daughter help, like, motivate what you do?
Yeah. Becoming a parent has definitely changed me entirely. I don't think you understand how mortally wounded you could be by something bad happening to your child until you actually have one. You just can't, it's just not something you can understand. Like, I now know that you could torture me by making me listen to my baby cry. I would psychologically be tortured. I would probably, you know, rip my hands off down to nubs trying to get to her. And so you think about that, and you think about how that's magnified across the parents all across the world. And, you know, particularly for me now, a lot on my mind is the climate, climate change, and desperate people in other countries, and how that's going to play out here over my daughter's lifetime. And, you know, it's heavy. I wonder every day if I'm doing the right thing in the world. Could I be doing more, right? I'm surrounded by an iPhone and 2 MacBook Pros right here. And I think about the rare metals that go into these computers and these iPhones and how we're mining them out of the earth. And I have probably, you know, 6 personal computing devices. So to do this science that's propelling us forward, I'm also consuming resources at an alarming rate, right? Personally, on a personal level. So...
But the science is important, and what you do is important, and I think the other thing too, that, you know, there's a lot of people doing science and working to advance us forward as much as possible. But I also think the science communicating is important, and that's, I think, a great reason that you're here doing this, is because we need people to be vocal and talk about these things. I think that's important too.
And I think a lot of people can honestly say they don't know a scientist. I think there's a lot of engineers, like a lot of people know engineers and a lot of people know people who work for corporations, but I don't think a lot of people know a scientist. But it has been very important for me to do these things as a way to force myself to explain things like Studies 5, but in hopefully a manner that really can connect with people. I think, it's what I've called, before with other people, the tyranny of jargon. You start using all these words and suddenly you're putting a wall between yourself and the people that you're talking to. And it's so easy for scientists to do because science really is another language made up mostly of Greek and Latin words. And, it literally is another language. But that's one of the ways that I try to connect with kids particularly from non-English speaking backgrounds. Because what I like to explain to them is that they already have a sense of the conversion of numbers from the name of the numbers themselves. So that was something that I didn't have going into science. When I learned that numbers had to be converted and that there was milligrams and centigrams and milliliters and, deciliters, I was just blown away because I couldn't understand it. How is it that everybody knows about this, but not us, and then I had to learn it, and it was really hard to learn how to convert numbers, the most basic thing. And so I like to really try to inspire my kids that, maybe speak Spanish at home, maybe they're English language learners themselves. And I like to point out all the root words that are similar so that they can know that they already understand the language and that's not a hurdle for them.
So it's that connection that I think is important, and having conversations about these things. And I think the more people that are out there, whether talking to kids or explaining it like Dan and I are 5, like the science behind things.
Right.
But I said, I learned things yesterday at the Children's Museum.
Yeah.
So
and we learned things tonight, and hopefully the people that have watched and will listen to this will learn things too. And I think that's all that we can hope for, is that the more informed people are about all of these topics, the less fear will be out there about them, and the more we can, in turn advance and not just fearfully, and make the world a better place, maybe.
Yeah, I know.
We could do it.
It's such a tall order. So
it's a tall order, but yeah.
Yeah. We went to visit the aquarium today, and there was a display about the acidification of our oceans. Mm-hmm. And it was a great display, but, I ended up having to, what does that mean? And so I'm kind of good at doing this, right? Thinking of an analogy on the fly. So I was like, the atmosphere has a lot of oxygen in it. It has a lot of carbon in it. And the ocean is basically like lungs are to our body. The ocean is a sink for this carbon, and our lungs, we breathe in oxygen, we exhale carbon dioxide, we regulate our pH in our blood by doing that. And the ocean kind of giant lung for the planet. So when the ocean is not, when it's taking on too much carbon because the water is warming, and that makes it easier for carbon to dissolve in the water, it becomes acidic. And then what it starts doing is that acidity, I think everybody has a feeling for what happens if you put a chicken bone in a can of Coke, which is very acidic, right? You start leaching out all of the minerals that make bone hard. And that's what's happening in our oceans right now. So you see a collapse of shellfish populations, which lots of humans are dependent on all across the planet.
Well, so you see, like, this is why you're a science communicator, because you've got the analogies, like, but that's
the analogies. Really, but the analogies.
It's what people need, though. People need those kind of explanations that you can connect to something that you are that you already know. And so I think that's where.
But I wanted to tell them, like, you need to change this display because nobody understands what the acidification of oceans means.
See, they need the analogies, but they need the things that make sense. But sometimes, sometimes you can give the people the things that do make more sense. But that's why we're very grateful that you came on to talk to us tonight about some of these topics that, words that we see out there didn't fully know. And this it's really helpful to kind of demystify some.
I wish people understood more about, because then they would understand that they can't actually change their blood pH by drinking alkaline water.
Oh, there's so much that people could benefit from knowing.
Alkaline it by adding an acid in lemon, citric acid to it. Somehow that alkalizes your body.
My goodness.
Oh,
does that make sense if you don't think about it?
It makes sense if you don't think about it. Just like so much of this makes sense.
Don't think about it.
Don't think about it. Don't think about it. Ignorance is bliss, and then go comment on the internet about how wrong all scientists are because they're paid by people.
And do it in all caps.
I get accused on a daily basis of being a shill.
Oh yeah.
And as we were talking about before we started recording, as we were talking about, no, none of us really make any money doing this, and we were talking about how
not making any money. I have no conflict of interest, because that's the thing that's what you're gonna get right now, is people are gonna be like, she is profiting off of GMOs. Or something, right? Or whatever.
Yeah. Oh, no, I know, because we've gotten it too. Because I know, making a film about, that is pro-science, biotech, vaccines, but no, I'm not getting paid by anybody to do it.
No.
Would have had a much bigger budget if I was, probably, but no.
I feel like what I am hoping for is that when people do get to know me, that they're going to see, there's nothing extravagant about my lifestyle. At times at times I've actually been really struggling, to put food on the table or whatever. It's definitely, scientists are real people.
Yes. I was just gonna, yes, scientists are people. And I guess to kind of wrap up, because I imagine when we're done, or when people are done listening, they will want to find your work on the internet and stuff. Where can people find you, and what projects, anything you'd like to promote as we close out here?
Yeah, so I think the best place to find me right now is on Medium. So I do a lot of writing for We Love GMOs and Vaccines. I love doing outreach for We Love GMOs and Vaccines. And I talk a lot about, biotech, GMOs, and vaccines. And I'm available for anything. So either on the We Love GV site, Facebook site, or you can reach out to me at my personal email address that has to do with the biotech company that I was the founder of for a few years. So it's Carol Curchoe at 32atps.com. It's all over the internet. You can find it anywhere. But yeah, it's, the method on Medium, or, the We Love GMOs and Vaccines Facebook page. I don't have a personal science communication thing set up. I've been toying with the idea of doing it, but
I think you should.
Yeah, and I think you should. Yes.
We could definitely share things and definitely get you some follows from at least for me, because we got just a few people that follow my page.
Just a few, yeah.
Just a few. But yeah, so we'll link to all of your information in the notes to the show. And thank you so much.
Fabulous.
I'm sure we will have you back to demystify some other, complicated topic for us at some point. So thank you so much for coming on today.
We will tell you what, we could have a joint show with her and Kevin Folta talking about strawberries and cows and making strawberry milk, because that's what he's working on. So
strawberries.
That's a crossover I can believe in.
Yeah, that's the future liberals want, I think. So
I know we talked about that like 5 minutes in. That's all I can think about now is strawberry cows.
Yeah, you're on that. Okay.
Well, but yeah, thank you so much for taking the time with us.
Yeah, awesome. Thanks for having me. Thanks to our listeners.
Yeah. All right. So Dan's gonna stop.